Okay, so now here we're on section 1.3, which is actually book section 3.1 and 3.2. And this is going to cover how the different bonds are formed to make the macromolecules. And I think I'm going to show a few animations first. This should actually all be review for you. But I'm going to show a few animations and then I'll go through the diagrams here. So this is going to cover hydrolysis and condensation and how that is going to be used to make all the different types of macromolecules in your cells. Okay, so this is going to show us how we form a triglyceride. Okay, I don't know if there's any sound here.
Triglycerides are made up of 3 fatty acids and a glycerol molecule. Glycerol forms the backbone of the triglyceride. In a process called esterification, fatty acids.
All right, so what you should be able to see and recognize before this even happens is that you're going to be able to take water out to form bonds. So H2O, see these H2O, and same thing here, H2O. It doesn't matter which O, because it's going to leave behind the O. So imagine what it would look like if you took out the H2O and you're left with what's left there on all 3 of these. So these are 3 different fatty acids, or it looks like 2 of them are different. So they could be saturated fatty acids, unsaturated, long chain, short chain, but they're going to connect through. I like to think of this as the formation of a triglyceride, like keys on a keychain. So the keychain would be the glycerol and then 3 different keys to form the triglyceride. They're going to be bonded with the glycerol molecule. So visualize the water coming out. Remember, that's going to be called condensation, water coming out, just like condensation is water coming out of the atmosphere. And sometimes that's called dehydration synthesis or condensation. All right, so let's watch this.
Joined to the glycerol molecule. The linkage between the acid group of a fatty acid and the alcohol group of glycerol forms an ester. Since water is released when a fatty acid links to glycerol, this type of reaction is called a condensation reaction.
All right, so this bond here is called an ester. It's going to be C, carbon, oxygen, carbon, double bond, oxygen. That's an ester linkage. And it's formed by dehydration synthesis or condensation, water coming out. That's the ester. Okay. So that's on triglycerides, but remember, it's going to be the same type of reaction whether we're making a protein, a lipid, a carbohydrate, or even an nucleic acid. All right, so let's see what else we have here for animations. All right, again, these are going to be review, but right here we have 2 glucose molecules. And we're going to take out water, and what's left here is going to be called a glycosidic linkage. Glycosidic linkage is going to be the COC.
A condensation reaction joins 2 molecules together to form one larger molecule. An enzyme removes a hydroxyl group from one molecule and a hydrogen atom from another, then speeds the formation of a bond between the 2 molecules at their exposed sites. Typically, the discard.
Okay, so this here is called a glycosidic linkage. Glycosidic, linkage. It's going to be a covalent bond. And if we wanted to, so this is actually 2 glucose molecules make something called maltose, like malt sugar, a disaccharide. And to break it apart, we can use hydrolysis to add water back in. Water, lysis means to break.
Discarded atoms join to form a molecule of water. Hydrolysis is a type of cleavage reaction. It's like condensation in reverse. An enzyme splits the molecule into two parts, then attaches a hydroxyl group and a hydrogen group from water to the exposed sites. The result is two smaller molecules. Condensation and hydrolysis are used to put together and break apart many biological molecules.
All right, let's see what else we have here. Cleave. I just have a few that are very, very similar to each other just to kind of hit home with it. So this is going to be exactly the same as the other one. Let's just look at it again. It's not going to hurt to look at it again.
A hydrolysis reaction separates a compound through the addition of a molecule of water. In digestion, disaccharides are hydrolyzed into two monosaccharides. Here, the disaccharide maltose splits into two glucose molecules.
Okay, and we could put those together by taking the water out. And that would link them together through the glycosidic linkage. But in that case, it showed you them breaking apart through hydrolysis to form two glucose from one maltose. The disaccharide into two monosaccharides. And this looks like it's going to be just a different showing the condensation reaction with two different monosaccharides, glucose and fructose. You might remember what that one is. 1 unit glucose, 1 unit fructose. Give you a second to think about that.
A condensation reaction joins two molecules together while releasing a molecule of water. The disaccharide sucrose is formed from the condensation of two monosaccharides, glucose and fructose.
All right, that is a glycosidic linkage. I just remember it because it's like usually glucose is one of the sugars that's involved. So that's how I remember that one's called a glycosidic linkage. Okay, form covalent bonds. So I think that's it for what I had for animations. Yeah, I covered them all. So I'm just gonna very briefly go through just a quick summary of that. So you have here two glucose units, take water out, that form that glycosidic linkage. Now we have the disaccharide maltose. We can hydrolyze maltose by adding water back in. Hydrolysis means to break using water, so we can form two monosaccharides, two glucose units from the original maltose. And this is the same. This looks like the same thing here. It's the same exact thing. And then that's nice because it points out that's a glycosidic linkage, so I don't have to write that out. You can see that. I said that like 10 times already, but hopefully you can remember that one is called a glycosidic linkage because it has the linkage when you have glucose there. It's the same thing when you're forming sucrose too, but I just remember it because it sounds like glucose. All right, same thing. If you remember, maybe if you don't remember this, if you didn't have me, but back in the day, in 9th grade, I had you write out amino acid general structure like, I don't know, 10 times or something. And then to take the water out and connect them. So you can imagine this H2O coming out here and connecting what's left here. And then this is going to be important when we talk about proteins. So let's cover it now while we can. This is a dipeptide, but dipeptides are not too common. Usually when we have amino acid chain, we're talking hundreds, even thousand in length. But there are some very smaller peptides, so in this case a dipeptide. That's not unheard of. So in this case, two amino acids, there's going to be two ends to it, and they're not going to be the same. So one end has got a nitrogen on it, and the other end is going to have a carboxyl group on it. So when you're talking about the structure of a protein, the amino acid, there's going to be one side that's called an N terminus, so the nitrogen end of it, and the C terminus, or the carboxylic acid terminus of the protein. The bonds between two amino acids are called peptide bonds. So it's always the same type of reaction, hydrolysis and condensation, whether we're making proteins, nucleic acids, lipids, or carbohydrates. So this is just kind of showing these, all three of these are the same fatty acid, some like really short chain ones. Notice, if you remember again from bio, you're going to have, this one is completely saturated with hydrogen. So these are 3 saturated fatty acids that are connecting to this kind of glycerol key chain by condensation. So the water comes out, and these are called ester linkages. And then this just shows you a few different carbohydrates, and you could see their glycosidic linkages. Cellulose, starch. You don't need to know the difference between them as far as their chemical components, but remember, cellulose is the structural polysaccharide of plants. Starch is the energy storage form of carbohydrates in plants. Glycogen is the energy storage form of carbohydrates in animals, and we have roughly about a day's worth of glycogen that's stored up mostly in your liver, but a little bit in your muscles. That's why you can go some periods of time without eating, that you're usually going to be tapping into first your liver glycogen, and then secondly into your fat stores and your lipid stores. Usually you don't tap into your glycogen stores of your muscles until you're like working those muscles out pretty vigorously. So that's a kind of cool thing, that it kind of stores those in your muscles. You don't use up the muscle glycogen unless you're using those muscles. And then chitin is a different polysaccharide that is very tough. It makes up the exoskeletons of insects and arthropods in general, things like lobsters, crabs. Chitin, but also makes up the cell walls of fungi. So it's a very tough polysaccharide. Okay, so that is all for that one. Hopefully this one was a little bit shorter. I'll find out when I go to stop this recording. And I hope that you have a good day.